Well, it's really a pleasure to be here and show you what we're up to. We do a lot with deep data for actually trying to improve healthcare. So if I were to ask you, how many of you have been wronged by the healthcare system? Probably all of you, or nearly all of you, would raise your hand and say, yep, that's happened to me at least once. And we think the healthcare system's broken in many, many different ways. First of all, we tend to practice sick care rather than really practice healthcare in the first place. And if you think about it, even when we do practice healthcare, it's a pretty archaic fashion. You typically get in a car to travel to a physician's office, and it's always at an inconvenient time. When you show up at the doctor's office, it pretty much looks like the same as it looked for the last 40 years or so. Maybe there's a few new gadgets in there. While there, they'll stick a needle in you, which can hurt and pull out a lot of blood. And from all that blood they pull and the time you're there, they actually don't make very many measurements. And then lastly, from those measurements, they'll treat you based on population averages. We think every one of these steps can be improved. And just to illustrate this last point about treating people based on population averages, you've probably been told since you're little that your temperature when you put a thermometer in your mouth is 98.6. But if you actually look at the data that are out there, this is a more accurate number. This is one particular study. It's more like 97.5 is the median. There's a recent study out of Stanford looking at 300,000 people. They say it's 97.7, but it's not 98.6. But the more important point is what's shown here. There's actually a spread. The 25th quartile is 94.6. The 75th quartile is 99.1. So what that means in today's world, if your normal healthy baseline is 94.6 and you go to a physician's office and they measure you at 98.6, they'll tell you you're normal, you're healthy, everything's fine. What are you doing here? Go home. When I guarantee if you're up 4 degrees over your baseline, something's off. You're probably quite ill. And so that's a big theme of our work. Know what your healthy baseline is so you can see shifts from that. So your health, I'm sure you know, it's influenced by many things. It's influenced by your DNA, and it's also influenced by your lifestyle and environmental exposure. So exercise, food, mental health, all basically can contribute to your health. In fact, it's thought your DNA contributes about 16% of your lifespan. We're in a world now where it's possible to quantify all of these, and we'll talk about some of this. You can quantify activity with wearables. Food's a bit clunky, but still possible. Same with mental health and environmental exposures. We actually have an exposometer we use for measuring some of these things. But even if you don't measure them directly, you can measure their effects by doing deep data profiles on people. And so a number of years ago, we set up something we call personal omics profiling, and it takes advantage of a revolution in technologies that's emerged over the last few decades. So many of you have heard about the DNA sequencing revolution. It now costs us about $350 to get a genome sequence. We order them actually, but pretty soon it's going to, the price of sequencing a genome for the reagents will be about $100. Now it's still going to cost a lot more to actually interpret the genome, but the actual cost of sequencing it will be very, very cheap. So all of you will be able to get your genome sequence. Kind of curious how many of you had this already. It's been a lot quieter, but there's also been a revolution in mass spectrometry where you can measure now tens of thousands of molecules out of people's blood and urine and get very deep profiles. These are the same devices they use in airports for measuring bombs, detecting bombs. And so anyway, we use them for trying to detect molecules in blood. We'll come back to that. And then lastly, there's been a revolution in wearables, these devices I hope many of you are wearing. We'll talk about that as well. And so this has enabled us to collect data at a level that's never been possible before. And so, as I say, a number of years ago, we set up this personal omics profiling. In fact, the study was started, well, on me over 13 years ago, but on the cohort as a whole, it's a smallish group of people, 109. We've been profiling them for about 11 years. But what we do is we'll sequence their DNA just once, but we'll actually sample their blood, urine, and poop, and we'll measure their as many molecules as possible out of their blood. So we'll measure all their RNA, their proteins, their metabolites, their lipids. The microbiome will measure out of actually your poop. Turns out that's a very important health measurement, as well as we measure your nasal, tongue, and skin microbiome as well. This is on top of very deep clinical testing. The wearables we brought in about 10 years ago, back when these were just fitness trackers, we realized they were pretty important health monitors. We also do some advanced tests like stress echocardiogram and glucose control measurements like insulin resistance, because it turns out I'm type 2 diabetic. And so we have a strong interest there. Excuse me. So basically what we'll do is we'll sample people every 3 months while they're healthy. It's not shown here. And if an adverse event like a viral infection comes along, we'll take typically 5 to 7 more samplings. And as I say, we've been running the study for about 11 years. So you might say, why are we doing all this? We're trying to understand what a healthy profile looks like, not what a sick one is. What does a healthy one look like? What happens at the first time of illness when we sample at those times? How does it change over time, your healthy profile? What's the difference between you and other people? How similar and different are we from one another? And important for the discussion today is, can these advanced measurements like genome sequencing, wearables, can these be useful for monitoring health? And I can tell you when we started this study, there was a very, very strong reaction to not sequence genomes of healthy people. Virtually all physicians said to me, Mike, that's a really bad idea. You're going to overload the system. You turn everybody into hypochondriacs. It's going to lead to millions of dollars follow-up tests. Well, that actually hasn't been the case. In fact, it does cost maybe on average $700 in follow-up tests. But in general, people are warmed up to the idea, because you'll see in a minute, many people have had life-saving measurements from their genome sequence. Anyway, with regards to using these advanced technologies, I'll just jump right to the chase. In the first 3.5 years of profiling people, we had 49 major health discoveries. So nearly half the people learned something pretty important about their health, all before symptoms, so pre-symptomatically. And it spanned a wide range of areas: hem-oncology, cardiovascular disease, metabolic disease, so on and so forth. And just to give you a few examples, some of these were a big deal. We caught someone with early lymphoma, 2 people with pre-cancers that can convert to aggressive cancer. 2 people had serious heart issues, one picked up by genome sequencing, another by wearables. And that's another pretty important point. No one technology found this. Sometimes it was a genome sequence, sometimes it was imaging, sometimes it was some of the molecular markers. And usually it was a combination of markers, said something was off, again, all pre-symptomatically. So the way we like to think of this is, what if your health is a 1,000-piece jigsaw puzzle? We're trying to take 500 or 600 pieces, whereas today in a physician's office, they'll typically measure 5 or 6. We're just doing much deeper profiles, getting a much clearer picture, if you will, of people's health. So just to dig in a little more detail, genome sequencing was very, very important. 12 of the first 70 people actually learned something pretty important from the genome. This is what I'm going to teach you a little genetics here, see if you can follow what I'm going to say. But many people can be at risk for certain diseases by single gene mutations. And a classic example is this one here. If you have a mutation in the BRCA gene, you're at very high risk for breast and ovarian cancer if you're a woman, breast cancer if you're a man, a bit of prostate cancer as well. And that's true for these others. They put you at risk. Single gene mutations put you at risk. And so by sequencing a genome, you can actually find these things. And so from the first 70 people, 12 had actually a pretty important mutation they found, including a BRCA case. And then we had 2 others, actually 3 others, who learned something pretty important that revealed something. So for example, one individual we sequenced had a mutation in this gene, which is a heart gene, if you will. Some mutations lead to cardiomyopathy. And sure enough, this person did a follow-up test and actually has a heart defect. His father died of a heart attack, actually. He's pretty young. So this discovery we think was quite valuable. Another person, also quite young, had a mutation here in this gene. There were 2 people who had this, and that puts them at high risk for certain kinds of cancer. They did a whole body MRI follow-up, and sure enough, they actually had early thyroid cancer that was removed. They were able to keep most of their thyroid, and so they don't need replacement therapy. So that was actually very valuable. And 9 individuals coming in the study were thought to be type 2 diabetic. One of those actually has a mutation here in this gene. And it turns out they're not exactly type 2 at all. They're a different kind of diabetics called MODY, and that has a different kind of medication you usually use. So they were on the suboptimal medication for many years and then switched after they got their results. So even though it's a minority of cases, 12 out of 70, if you're one of these cases, it's a pretty big deal. And as I say, in some cases, we think it was potentially life-saving. Now, this is what we call single gene mutations, often referred to as Mendelian genes. Most diseases are actually complex disease. And sorry, a slide got lost here. For complex disease, they're not thought to be due to single gene mutations. They're thought to be due to common changes we have. We all have, changes in our genome. We'll have 4 million relative to the reference. So it's a lot of changes. And then most of them are quite common. They're just natural variation of the population. And so for complex disease, the way that's thought to work is that people are thought to inherit these common variants. And if you get a lot of the bad ones, you're at higher risk. And if you get the good ones, you're at lower risk. And so that's how most diseases like coronary artery disease, diabetes, things are thought to happen. So we've come up with whole new ways of analyzing the genome with AI. Actually, it's people have used something called polygenic risk score, which doesn't work very well and doesn't really get at the genetic basis of most diseases. And I'll just summarize by saying with that, by using even before it was popular, we started using AI as a new way to analyze the genomes, which turned out to be very powerful. So we were able to go into something called abdominal aortic aneurysm, which is semi-common. 10% of people over 65 die of this. And the way you know you're at risk for this is your aorta bursts and you die. So it's a terrible way to know you're at risk for this. And so we think getting a genetic test will be powerful. And so with our new method, we actually went from 3 genes that have been identified to find 60 genes. And we now have, I'll show you in a minute, a predictive model for actually AA, which is quite powerful. Here's another one. I'm sure you may have heard of Lou Gehrig's disease, ALS. There were 7 genes identified from genetic studies. With our new AI method, we actually found 700 genes and could explain 6 times more heritability. These are all known to be heritable genes or heritable diseases, and we were able to find them. And severe COVID that can happen in young kids is also heritable, it turns out. And we went from 47 genes from a very interesting study that had been done and found over 1,000 genes. We could explain 77%. So I know that's kind of glossing over this, but the point is with these new AI methods, we can get at the genetic basis of disease at a level that's never been possible before. And I'll show you for AA, that's turned out to be powerful. We can set up a very predictive model for based on genetics, whether you're at risk, and we add a little additional information besides your genome, we can get reasonably predictive about your risk for AA, which is kind of powerful. So the other thing we discovered is that by profiling people over time, we can actually see how they age. Remember, we're following, actually, it turns out, large numbers, tens of thousands of markers, and about 600 of them change over time, which we interpret as aging. And what we discovered is that everybody ages differently. So this person here, their top biochemical pathway that changes is their cardiac hypertrophic signaling pathway. So they're a cardioager, a heart ager, if you will. And we later learned this person's stage 2 hypertensive. This person over here is pretty typical ager. Their coagulation immune pathway, which is what this has evolved in, metabolic pathways all go up over time. We think we need as little as 4 to 5 measurements within 2 years, and we can tell you how you're aging. And as you say, everyone ages differently. We classify people into what we call ageotypes. And we were empowered to find 4. We know there are many more than that. But each column here is a different person. Each row is an ageotype. And so the 4 people over here, they're aging in all 4 categories. This person's aging in 3 of the 4, not much of a kidney ager, but ages in other categories. This person's kidney ager, not much else. This kidney and metabolic and so on. And this information we think is quite powerful because there are clinical markers associated with each of these ageotypes. And you can actually take action. You can change your lifestyle. I'll come back to this at the end and shift your ageotype in a beneficial fashion, we think. So we think by getting this information, you can actually make adjustments that we think can improve at least your biological age. Not going to change your chronological age, but you will change your biological age. One question I get asked a lot is how are you going to scale this? This is a research study that costs millions of dollars, which is true. And I'm in Silicon Valley, so the way you scale these things is, of course, you form a company. I'm a believer academics. We're great at discovery and proof of principle. We're not very good at scaling. So we formed this company called QBio. It does a medical version of what I just showed you. It doesn't do all the things I showed you, the ones, but we figured out what we think is most cost effective. And it's a deep medical grade test, and also does whole body MRI. Now, if you ask any physician today, should you do a whole body MRI? 100% of the time they'll say absolutely not. And if you ask Mike Snyder, should you do a whole body MRI? I'll say absolutely you should. And what's the difference? Well, physicians don't want you to do a whole body MRI for two reasons. One is it's expensive, that's true. But the other is that they're worried if you do a whole body MRI, you'll find nodules. And that's also true. 100% of us have nodules. And men have them in their prostate, women in their ovaries, 100% of the time. But my point is that that's not the point. The point is not whether you have nodules. The point is, do you have any growing nodules? And the way you know that is through these longitudinal measurements, follow people over time. And so just from the first 100 plus people, we found some pretty important things. We found some of the early ovarian cancer, which is rarely discovered early, prostate cancer, even pancreatic cancer, which is almost never discovered early. We found that through this longitudinal profiling, cardiovascular disease, some other things. So these we think are a pretty big deal. Once again, these are all found pre-symptomatically. We could detect these things before people had symptoms. And again, it's because we're doing deep data dive. We're getting a much clearer picture of people's health. And so this is how we're trying to scale this. So we'll be expanding this out beyond the Bay Area at some point. All right, so that's the general deep data dive on things. The other area we've been working very hard on is this idea of remote monitoring. Why do you go to physician office all the time to get measured for health? And you will still need to go see physicians. Don't get me wrong. We're not trying to put them out of business, just the opposite. We're trying to improve what they can do. But it's true that for a lot of measurements, you don't really have to go to a physician's office, and they're not really the best place to be measured in certain things. So we've been pushing two things. We're trying to, this concept of Amazonizing healthcare is really what we're trying to do. And we're doing it two ways. One is with wearables, and the other is home microsampling, where you take bits of drop of blood, mail them to us, and we'll actually do deep profiling. And I know what this sounds like, but ours actually works. All right, so wearables, I'm not sure how many of you are wearing a smartwatch or ring. I myself normally wear 4 smartwatches. If you look up, you'll see my various watches. One broke, so I'm down to 3. I have a ring that measures. Even my hearing aid, believe it or not, it is for hearing, but it's a sensor. It can measure physiology, and the companies think they're pretty good for that. So the reason these are powerful is they're measuring you 24/7. They measure you all the time as long as you keep them charged. So they're always tracking, and they're tracking some pretty important things: resting heart rate variability, skin temperature, your blood oxygen. They can even measure your conductance on your skin, which turns out to be valuable for certain things, as you'll see in a minute. So we started, as I say, using these on our cohort back when they were just fitness trackers. We thought maybe they'd be powerful health monitors. And what we discovered right away is that you can tell when someone's getting ill from an infectious disease from a simple smartwatch. And this was actually first picked up actually on me when I discovered my Lyme disease, of all things, with a smartwatch and a pulse ox, which measures your blood oxygen. Most of you have probably tried one of these at some point. And I just happened to be on an airline flight. I noticed my blood oxygen was abnormally low. It drops on airline flights normally, if that's true for everyone. But it dropped abnormally low, never came back to normal. I saw my heart rate was up. That's sort of shown here at low resolution. I saw it was up. This was all pre-symptomatically. And then I later got symptoms off and on. I did go to a physician in Norway. I warned him it might be Lyme because 2 weeks earlier, I had been in rural Massachusetts putting up fences where 55% of ticks were Lyme infested. And he wanted to give me penicillin, actually. He drew blood and saw my immune cells were elevated. He wanted to give me penicillin. And I said, no, I need doxycycline. It got a little bit tense there, as you might imagine. He did give in and gave me the doxycycline, cleared it up right away. It took it 2 weeks. And when I got back, I got tested. And sure enough, I was Lyme positive. I have antibodies. to the Lyme antigens, and I also had even some Lyme antigens or proteins around. And it's a well-controlled experiment because you can tell I measure myself a lot. I given blood before I left and was negative, so I seroconverted during this time. So the point out of all this is that I could first tell when I was getting Lyme with a simple smartwatch and a pulse ox. And so I won't show you the data for this, but we went on to show I had 2 years of data, and I was ill 4 times, and we looked at my watch data, and every single time I could tell I was ill from my smartwatch. My resting heart rate jumped up in advance of symptoms. And one was the Lyme case, 2 were viral infections. The 4th, I was even asymptomatic, but I was basically ill because I had high something called C-reactive protein, which is a sign of illness, quite high. So every single time we could see this, so we wrote an algorithm then to show we could do early detection just based on resting heart rate. So we published that in 2017. And of course, all of you know that in March 2020, at least in the Bay Area is when we got hit with COVID. You guys actually were the first to detect it up in Washington, in the US. And the bottom line is that, you know, waves have been coming. This is data that I think I stopped in 2022, and we're just hitting a big wave now that's kind of, maybe it's on its way down, but it's still pretty high. And if you think about how we measure COVID now or tell if people are getting sick in general, it's with temperature, which is a 300-year-old concept and doesn't work very well for COVID because half the people don't get a fever. So it doesn't really make a whole lot of sense in this particular case. There are, you know, these UV lights, and that one doesn't even make a whole lot of sense sometimes too, because when it's cold out, I don't know about you, but they'll shine on my head and I'll measure 33 centigrade, which is way too low. They weighed me in anyway, even though it's a worthless measurement. So anyway, we should be thinking ways around this. We do antigen and PCR. They are better, but you don't do them all the time because they're, you know, kind of inconvenient and a little bit costly. And so we think wearables and smartwatches could be very, very powerful for this. So when the pandemic hit, right away, we partnered with Fitbit, got 5,300 people enrolled, and 32 of them had COVID while wearing their Fitbit, and they had a diagnosis day and a symptom day. So we said, all right, can we detect COVID with a simple smartwatch? And the answer is yes. This is resting heart rate. It's plotted for the aficionados as a standard deviation plot. Just think of it as a resting heart rate. Here's the diagnosis day here in purple. Here's the symptom day here. But you'll notice this person's resting heart rate jumped up 9.5 days prior to symptom onset. You can't miss that signal. And it turns out we could detect this in 80% of the time, 26 of 32 people. And the median time of picking up the signal was 4 days prior to symptom onset. So it works 80% of the time that we'll detect it before or at symptoms, and the median is 4 days. So it's pretty powerful. We wrote, I'll show you in a minute, a real-time detection method where you follow people's personal baselines based on a circadian fashion. And if they get a jump up in heart rate for too high for too long, it actually sets off red alerts. By the way, you can sign up for a study here. We should put this in the chat. I'm going to send you my email in a second so you can get it from there too, but maybe somebody can put that in chat. Bottom line is with this real-time detection, again, it tracks you. So we built this thing. It's a cloud-based system. It can follow millions and millions of people. And so I'd love to have you all sign up. And basically what happens is if your resting heart rate, and it's more than that, signal, we want to pull in other data types too. That's why we need everybody to sign up. We want to perfect the algorithms. But basically, if you get a jump up in resting heart rate, a little bit off in sleep and steps, it'll give you a red alert. And it works same because it's prospective. It works. The median is 3 days prior to symptom onset. And here's our very first case. It's actually a typical case, it turns out. So this person had symptoms here. They were diagnosed positive the next day, and they were getting red alerts for 3 days prior to symptom onset. So same thing. It works 80% of the time, and it works for Apple, works for Fitbit. It should work for all devices. And it works here even in asymptomatic cases, same number, 80%. So here is a Fitbit case. This person was diagnosed with COVID here, and they were getting red alerts for 2 weeks prior to this diagnosis. So again, it's a very sensitive assay. We think you need as little as 2 beats per minute, and we can tell if you're getting ill. Now, it's not specific for COVID. So I got to tell you, it's more of a stressor alert in general. So certain things like intense exercise, you run a marathon, will your heart rate will be up for several days if you run a marathon. Not a regular jog that won't trigger the red alerts, but or even a regular run, but something super out of the ordinary, and other things can do it too, including other infections. The number one trigger of red alerts is workplace stress. Again, it's a stressor detector. I know as we pull more data now with respiration, we should be able to tell the difference between workplace stress and a respiratory viral infection. To me, that's a no-brainer. So anyway, we're trying to protect this, get it working for basically all kinds of conditions, including mental health. Actually, we have some studies running there. And as I say, this is set up for millions and millions of people. Love to have you all roll. It turns out it's more sensitive than an antigen test. And I'll show you my personal story, which is I was, normally there are green days, but I had a particular family stress I had to travel for. And so that set off red alerts. And then I was back and I was getting ready to go to New York City. And I woke up one morning, I'm just a little bit congested. And I'm wondering, is it allergies? Am I coming down with something? So I do an antigen test and I'm negative. That's the test spot. I look at my smartwatch and I'm positive. So what do? I got on the plane and I went to this meeting in New York City. And before you can go to the meeting, you actually have to get tested. And once you know it, I'm bright positive. So I spent a whole week in a tiny little hotel room in New York City, all because I listened to my antigen test and not my smartwatch. So again, it's very, very sensitive and can detect these changes. Now, it's obviously not as specific. I didn't know I had COVID or I never would have got on the plane, although I won't get on a plane again when I see this sort of thing happening. So, yeah, so we think it's very powerful. And this isn't just me. We've had several people in the exact same situation. They turned positive by watch before their antigen. It's just very, very sensitive. All right, we even think you can detect long COVID. I won't have time for this, but you can pick this up because there'll be a signal for those who get long COVID. They'll have elevated resting heart rate and other things as well after they get COVID. Now, we think this is the tip of the iceberg. We've used machine learning and AI to be able to actually predict clinical markers from features in a smartwatch, meaning your smartwatch, if you think about it, it's very powerful. It's shining light on your blood and looking for spectroscopic shifts. That's how it picks up oxygen, blood oxygen. And so you can actually, it's really a very sensitive instrument. And so we can pull features, meaning the blood oxygen, all these things from your smartwatch, and use them to make predictions about clinical measurements like red blood cell count, hemoglobin levels. These are important for anemia and other situations. Hemoglobin A1C, fasting glucose, important for diabetes. We even get a signal from that. So these devices are very, very powerful. These aren't clinical grade measurements, but they're good enough to see if you've got a shift from your baseline, if you get it shifted from your baseline, and you should be able to pick that up again in advance of symptoms. So we think these are going to be powerful health monitors in the future. The other area we've been pushing very hard is glucose monitoring. And I don't know if any of you have used these, but they're very, very powerful devices. And just to our claim to fame is they were being used for type 1 and insulin-dependent type 2 diabetics. These devices will sit on your arm or you put them on your stomach, either place. And what they'll do is they'll measure your glucose every 5 minutes, your glucose levels, obviously critical for diabetes. Our claim to fame is we started putting these on normal and pre-diabetics. And we discovered that some people are normal. That is, they have good glucose control. They don't spike much even after eating cake and such. Other people are moderate spikers, and other people are severe spikers who are just as bad as diabetics, even though they've been classified as normal. And so we wrote algorithms that classified people into glucotypes based on their glucose levels and spiking. And some people are low, others are moderate, and some people are severe glucotypes. The other thing we discovered is that different people spike to different foods. And Aaron Siegel had found the same thing. That is to say, some people spike to potatoes, others to pasta, some to white breads, and the brown bread. Everybody's different. And to show you some examples of this, this is from Aaron's work. This is an individual who spikes a banana, but not a cookie. Probably the only person on the planet not to spike to a cookie. This person spikes a cookie, but not a banana. So that's more typical, by the way. So again, people spike to different foods. We had found that some people spike to bread and peanut butter, probably from the bread. Others have protein bars, which have carbs in them. 80% of people spike to cornflakes and milk. If you don't remember anything when I talk, keep away from that. This stuff's evil and poison. So it's not good for you, I think. It could be worse than smoking. So anyway, we think that's bad. All right. So I tell you, one of the things we discovered is that even wearing, this is from my company, January Eye, even wearing a glucose monitor for 10 days, it's very eye-opening. You will improve without even trying. Just this is doing a food logging and wearing a CGM, not even a recommendation program. People improved what they call their time in range, or they're more optimal glucose levels. Nearly everybody improves it, healthy as well as type 2 diabetics. So again, these are very, very powerful devices. And they've gone on, they have a 32 million food database. They have a whole program around this. I'll give you a QR code for anyone interested. I forgot to say at the outset, I'm 100% conflicted in everything I'm telling you. So you can take what I say with a grain of salt if you want, but we think this stuff is powerful. The other area we've been pushing very, very hard on is this home microsampling that I mentioned before. So we spent about 7 years on this. And the idea is you take little pricks of blood, either from your finger as shown where this device collects from, or you can actually, the devices we use now are from your shoulder. And they're actually, especially ones on the shoulder, are pretty painless. They're collecting very small amounts, really tiny droplets of blood that you then FedEx to our lab overnight. And we'll run them in these various profiles, looking for all your metabolites, all your proteins, all your lipids, do deep, as well as targeted assays against some pretty important molecules, proteins and metabolites. In the end, we're measuring about 2,200 molecules in people's blood. We actually spent 7 years perfecting this. The key was to get the molecules stabilized. So I'll tell you, in the end, it worked well. Most of these are pretty stable. Some of the lipids are off, but in general, we know which ones and we just ignore those or correct for them. So then we did some really fun studies, and I think you guys might enjoy this. We had 32 people drink the shake that you can buy in CVS called Ensure. Many supermarkets carry it too. And basically, we sampled people either before they drank the shake or at 30, 60, 120, 240 minutes after they drank the shake. And we looked for changes based on this microsampling and mass spec and these assays I mentioned. And what we discovered is that each column's a different molecule, and this is a time series. Bottom line is hundreds of molecules change after drinking the shake. That wasn't a surprise, but we could measure some pretty key molecules like insulin, C-peptide. This is a famous molecule called incretin. This is what Ozempic and these drugs are trying to mimic. Basically, they're very powerful. Anyway, we can measure these and we can follow their kinetics. And what we discovered, here's the cool result. We had 32 people who drank the shake, got profiled, and everybody responded to the shake differently. So each box is a different person, and this is the profile. And this is just showing 3 carbohydrates. Again, we're measuring over 1,000 molecules here. And what we discovered is that, again, people react differently. They tend to behave the same as a class, meaning this person, their carbs plummet after drinking the shake. For this person's, the carbs skyrocket, up, up, up. This person's flat, this person's goes down, this person's goes down. So everybody's responding to shake differently. We could then classify people into 5 types based on their patterns. And I'm showing that here. Gray is the average of the group, and each colored dot is a type of molecule. So these are the inflammatory markers. That's the one I want to tell you about. So for this group of people, when they drink the shake, their inflammatory markers goes down. Yet for this group of people, when they drink the shake, their inflammatory markers goes up. Same with this group of people, up. Same shake, anti-inflammatory to this group, pro-inflammatory to others. So if you think about 10% of people having inflammatory bowel syndrome, we think this is a big deal, meaning we can see what's causing inflammation to people. And so most people who have inflammatory bowel syndrome have no idea what's causing, or irritable bowel syndrome, I should say. And so we think actually knowing this could be very, very powerful, and it'd be easy to measure with the microsampling. The other thing we've done is shown right here. We've done very, very dense profiling, combining everything I told you. So this is an individual who basically gave samples every hour, every waking hour, for 7 straight days, a total of 98 samples. At the same time, was wearing this continuous glucose monitor, as well as a smartwatch, and doing food logging and tracking activity. And this part's not a surprise. Once again, we see over 1,000 molecules changing, and we do something called time lag correlation. We're trying to see which molecules and physiology correlates with each other. And by doing this time lag correlation, you see which events are upstream of others. And the idea is that upstream events might be more causative for downstream events. So what we did then was we did this correlation, and what we discovered, just correlating with heart rate, glucose, and steps, we found thousands of associations. And right here are the lipids in this purplish color. Green are the metabolites. These are the lipids here in blue. And what we discovered, again, is lots of associations, and some are obviously made sense. So, for example, insulin C-peptide comes up after your glucose elevates, but we can measure. We can say it's exactly 10 minutes for this person. We discovered a new inflammatory marker that goes up after glucose. That was kind of cool. And this might be responsible for why diabetics have a lot of inflammation, we think. And then biggie is here. We actually discovered, this is a new finding, alpha-synuclein is actually showing an interesting pattern. We think it correlates with stress. Now, alpha-synuclein has been associated with dementia and Parkinson's disease. So what we imagine is that if you're a high risk for alpha-synuclein, you probably want to know this, what stress is, what triggers this to go up, so you can avoid that and keep it lower. And I'm not saying it would prevent dementia, but it should be able to push it off for a few years, just like for diabetes. By keeping your glucose down, you should be able to manage it better and push off your diabetes. And we think the same can be true for some of these neurological diseases. So that's an area we're pushing very, very hard on now. So the bottom line out of all this is that we can track activities at a personal level and see how people, what kinds of behaviors and lifestyle things cause what to your biochemistry. So to better be able to do this, we built a dashboard, much like your car dashboard, that pulls in your information, that pulls in your wearable data, your omics data, your clinical data, and presents it in a fashion that you can follow at whatever resolution you want, daily, weekly, monthly, what have you. So where we think all this is going, this is Mike Snyder's world. It may not be everyone's world, but I can envision a world where people get their genome sequenced before they're born, and that's now theoretically possible. It's not routinely done, but it could be. And then together with omics measurements, as well as, so these detailed biochemical measurements, as well as wearables, we'll be able to better track people's health. In fact, we'll have personalized AI tracking things. We have one built around me already for predicting your risk, catching disease early, again, ideally pre-symptomatically, and then monitoring and treat diseases. So as I say, one of the things we do here is we spin off companies as a way to get this out to the broader group. And as I mentioned earlier, I happen to be fortunate. I've been able to co-found 17 companies over the years. And they generally spin off, all the ones in boxes spun off from the study I just told you. There's a genome sequencing company here. Here's the big data one I told you about. Here's our continuous glucose monitoring company. I feel free to sign up for this. You can write to me, and this is, send that. We have a watch company. This one's cool. I'll tell you about this a little more in a minute, where you do the microsampling. You take a drop of blood, basically, you mail to us, and we'll give you back a profile of 500 metabolites related to health. Here's a long COVID company that does the microsampling and some front-end data decision making as well, data collection and decision making. So again, this is our way of trying to get what we discovered in the lab out to the greater population, and they can do it just 100 times cheaper than my lab would ever be able to do it. This one here, I believe, is Rhythm, and this is the yellow one. And so to dig in this a little deeper, this yellow one does the microsampling that you do at home and mail in, and they'll measure over 20 different wellness categories, if you will, metabolic health, inflammation, so on and so forth, oxidative stress. These are all a big deal. And they'll even follow your longevity and the age of types I mentioned before, so they can track your biological age, if you will. So you may know you have an age in years from when you were born, but your biological age could either have you older or younger. For this particular person, they're a little bit older than their biological age. And when you dig in why that's the case, well, some of their heart markers are up relative over average, and their metabolic markers, in this case, just a touch up, and their inflammation markers are lower. So they have AI-based recommendations to be able to help you improve this and improve your wellness in these categories. with the goal of improving your age. And actually 95% of people do actually improve from the first measurement to the next one, based on lifestyle changes. So this is the group that actually did all the work. Again, I've been very fortunate to be able to work with this amazing team of people who's been able, as I say, to try to bring these new technologies to bear on transforming healthcare. And so I won't have time to go through them, but this is our wearables team when it was at its peak of COVID. These days it's much smaller, but still powerful. We're measuring all kinds of things around this chronic fatigue syndrome and other stuff. I don't have a sign-up sheet for our studies, but you can email me and we'll send you one. Or I guess the host can send it out. And this is a team that did the microsampling, the home microsampling. So that's the story I wanted to tell you. Once again, thank you very much for having me here. Happy to take questions if you have some. So.
Hello, name's Damien with SAA. I'll be asking some of your more pressing questions here and the questions and answers here. Thank you, Dr. Snyder, for such a interesting and informative talk. You definitely have the Q&A section here jumping. So I'll go ahead and kick off our first question coming from Philip Sue. Just out of curiosity around privacy concerns, who would own the data that is collected, and how do we prevent the data, particularly our genome, from getting into the wrong hands?
Yeah. Well, who should own the data? You, of course. I think the person should own their own data and always have access. That's easy to say. It's not always so easy to get, even from your current healthcare provider. So I think you should be in charge of your own data, and I think you should have the decision who can share it. I do think that there is a benefit to sharing data. Privacy, I know, is a big concern for a lot of people. I'll be perfectly blunt. Nothing is totally private. As I'm sure you know, everything is hackable. So we certainly take every possible precaution. We have everything encrypted to be able to keep things as private as possible, as do most companies. But there's never a 100% guarantee that you won't get hacked. So far, we've been, knock on wood, very fortunate. I would hope that you not be discriminated against based on your health information. So far, I've not seen any evidence of that. There are rules that protect you around your genome sequence, meaning genetic information cannot be used to discriminate against employment or healthcare. It can be for long-term disability and life insurance. You can argue whether that's suitable or not, but the bottom line is, I'm a believer that as a wealthy society, which I believe the U.S. is, everyone should have access to some minimal health level of healthcare and not be discriminated against. So I would hope that no one would ever get abused by this. And certainly the power of sharing the data is so powerful that we really do learn a lot because people do share. Our group, by the way, has agreed to make their data completely open, the majority of folks, meaning their genome sequence, and none of them have ever been abused. And I have 2 petabytes of data just on me. It's all out there. You can download it and play with it if you want. Nobody's abused. I've gotten some interesting comments back on it.
So thank you. This question comes from Alicia Tozer. Can you elaborate on your thoughts on scalability, commercialization hurdles around data privacy or physician adoption for extended access to the broader population?
Yeah, I think we're certainly capable of getting it out to everyone. And I think inexpensively, like the yellow tests and the January AI ones, that's the CGM is the latter one. The first one's the metabolic microsampling. Those tests are in the order between $250 and $280. They're affordable, certainly to this crowd. A smartwatch, by the way, even a cheap one, for a $60 smartwatch, you can measure everything I told you about, heart rate variability, heart rate. You can go a long ways with that device. So I actually think that can get out to the entire planet, and I'd like to see that happen. Now, what's the barrier to this, I think, is partly what you're getting at. And the answer is actually the payment system we have. Our healthcare system's broken because it's sick care. Nobody pays you to keep you healthy. People usually go to the doctor when they're ill. I think you should get a discount if you wear a smartwatch, or if you do get measured more while you're healthy, because you'll probably do a better job at keeping yourself that way. You should get a discount if you get your genome sequence. So I think we need to align the financial structure better so that you are keep, so we can keep people healthy and extend their health span. I think the single payer systems like Canada, UK, places like that are a big advantage over the US, which is very fragmented. And nobody wants to put a lot of money into you when 18 months later you're going to move over to a new health plan. So we need to fix the system. We need to get our financial incentives aligned to keep people healthy. And that's another area. That's probably the number one area where the system's broken.
This question here for you, and I'm going to +1 on this one, from Julie Fiedler Armstrong. When you say cornflakes are bad, does this mean, or does this apply to all breakfast cereals, or is this something specific to just cornflakes?
Well, I don't think it's all breakfast cereal, but cornflakes and milk, I think it's the fructose in the cornflakes. So yeah, I think, but you probably want to measure, and to be honest, it's going to differ for different people, but even oatmeal will spike you. Not as bad as cornflakes, so. But and generally oatmeal has a lot of fibers, physium hus and things, metamucil, that's actually very healthy for you. It feeds your microbiome, keeps your colon very healthy. So there's a lot of benefits to certain kinds of cereal, but I would watch out those sugary ones. That's like a death signature. And I forgot to say this, but you may or may not know that 9% of the US is diabetic, 33% are pre-diabetic. Those numbers are just going way, way up. So the diabetes endemic is way worse than the COVID pandemic. And so we really have to get this under control. I think the CGMs, the continuous glucose monitor, is going to be a great way for that to happen. If any of you have ever worn one of these things, you'll know they're very, very powerful. You'll see exactly what foods spike you, which ones don't.
Okay. And this question from Sarah Demori, and apologies if I got your last name wrong, but how do you think in time this may be adopted on a population level, especially in countries with state-sponsored healthcare?
Yeah, great question. I think the wearables we can get out quickly to everyone, and some of that's already happening. The other kind of profiling, we have to show that these are, health systems want you to show that they're efficacious and that they save them money. So I think as we do that, I don't think the efficacious part's going to be very hard. I think the who's going to pay is hard. So I think the single payer, as you said, is the way to go. And I think we've got to get the financial incentives aligned. Once again, give people a discount. Get providers to give a discount if you are wearing a watch or getting sampled a lot. Don't wait for them to show up when they're ill, because once people have symptoms, it's very hard to reverse that. Like cancer that's metastatic and cancer with symptoms is a very, very hard thing to cure. Whereas catching cancer early, it gets cured. It depends on the cancer, but for most cancers, most of the time. So I think catching things early, keeping people healthy, it's very, very important.
Absolutely. And Trudy out there would love to know, is heart rate heart rate variability a good predictor?
It is actually. It's a really great one. We were not able to integrate that in our first study because we weren't able to pull the data in. It's what we're trying to get in now. Turns out heart rate variability is one of the best measures for health. Your heart rate should be variable because, and this gets technical, but your parasympathetic neurons are feeding in, and that's what actually triggers some of the variability. And when you get ill, the variability drops for all kinds of illness, for heart issues even as well. And cancer, all these sorts of things will drop your heart rate variability. So it's good to keep it up. It's great to follow your heart rate variability. As I say, we're not pulling in real time. When we do, I think it's going to make our sensitivity go way up for when people are ill.
Absolutely.
Great.
Are there any wearables out there that you recommend, or thoughts on the Aura Ring or the Aura Ring?
Yeah. So I get asked this a lot, and I have to say it depends on your lifestyle. So if you're not good at keeping them charged, get a Fitbit or a Garmin, because you can charge them for 5 or 7 days. You'll never miss data. It's wonderful. The Apple Watch is higher resolution, but does not charge for as long. And so that might, you miss data. And a lot of people charge it overnight, which is bad because that's actually when you want to do health monitoring. So so it depends. If you're really good at chart keeping your watch charged, they say get an Apple Watch. So, and then of course there's my company's watch, Omics, which of course is the best one. So feel free to, that one's not direct to consumer though. You'd have to be in a research study for that. Anyway, that has certain advantages. It's very high resolution and the data can come without going back to the company. So anyway, it's one of the reasons we set it up in part to be able to retrieve data. It's actually hard to get data from these companies at high resolution.
So, Bev out there asked, a tiny drop of blood or two, over 2,000 tests. I mean, it feels like shades of Theranos here.
You got it.
How's this possible?
No, Theranos could have done this. I should point out, we're not measuring LDL and HDL, some of these things that are clinical tests. What we measure are complements. But we can do a deep profile. Theranos could have done what we did. Now, we did spend 7 years, like I say, trying to get the right matrix to keep these things stable so that we get good, accurate measurements. And we are getting that. It's and we're not trying to, our mass spec is big. We're not trying to build something that's a tiny little box, which was another thing that I think Theranos is trying to do. So the bottom line is and also there have been advances in mass spectrometry to be able to do this. So it is real. We publish our data. We put the data out online. So any of you can look at it and download it. So we're not making it up. You can see it. And feel free to try it out. We'll give you your results and you can tell us, but it is also non-standard tests. A lot of these things are on the FDA site, but they're not necessarily normal clinical markers because we're measuring people very, very deep. So anyway, it's a very complimentary kind of assay. It's real though. It's not fake.
Absolutely. Kirk would love to know, you mentioned the desirability of whole body MRIs in part because of detection of nodules and then looking at ones that grow. Does that mean repeated whole body MRIs to detects the changes? Assuming so, how often?
It does, yeah. How often you should measure someone, nobody really knows. We're recommending every 6 months, but we don't really know. That's just a number. But you definitely need the longitudinal ones. And I'll give you a good case. I've had 18 now over the last, I guess, 7.5 years. And I have 9 nodules. I know exactly where they are. And I know none are growing. One's on my spleen, one's on my spine, etc. They're there, they're solid, and they're nice. Or well, they're not nice, but they're there. And they're benign. But I can show you, give you a good example. A friend of mine woke up one day and his arm wouldn't work. And they did follow up. They looked at his spine, and sure enough, he had a big nodule growing there. And the operator got it out, irradiated it, took care of it, and everything was working. So, and as a follow-up, then he did a whole body MRI, and there were 3 other nodules on his spine. So then the question was, well, were they there to begin with, or had his original cancer metastasized? And he didn't know because he had no baseline. And that's why I think it's really, really important to have a baseline so that if something, hopefully nothing bad will ever happen to you, but if something does, you would know what your baseline looks like, you'd be able to see. And I'll be honest, people live a long time now. It's estimated that everyone will get cancer in their lifetime, not necessarily lethal cancer, but you might get some slow growing thing that can treat and be taken care of. And so A, you better catch it early. B, you better know what your baseline is so that you know how to catch it early. So that's me on my soapbox, preaching out. But I feel very strongly about it.
So, and I think we have room for one more question, maybe 2 here. But are there any clinically relevant microbiome findings yet? What about, excuse me here, exposame? I don't know. I've never heard that word before.
Ozamic, yeah.
Yes.
Yeah. On the microbiome, there's one company called Day 2 that uses it to help explain what you might do for your eating habits. Your microbiome explains about 21% of your glucose variability. So it does have some predictive value, but that's the only thing. Everybody knows the microbiome is important, but has not yet made it as a clinical measurement yet. People are working on it. But there is one company called Pendulum. Here I have no conflict that actually they figured out a microbe that you've actually taken. It lowers your hemoglobin A1C by 1%, which is pretty good. So there are yeah, so it is important to keep your microbiome up, eat a lot of fiber, that's for sure, but it hasn't exactly worked its way as a clinical test. On the exposometer, same thing. This is now we're using it for research, we're measuring molds, all kinds of things. Lots of great results here. You might be alarmed to see there are carcinogens everywhere and plastics everywhere and all that stuff, but there are more in some places. Now the location is the number one driver of your exposures. So that actually we're selling as a research device. At some point, well, I don't know if it'll ever become a clinical test, not measuring person, but we think it's going to be pretty valuable for people.